Okay, welcome back, students. This will be our final presentation on the topic of nucleic acids. Please have your PowerPoint notes ready and let's get started. As I was mentioning, the information that is stored in the form of DNA has to be converted essentially into a new language, which is a language of peptides. So that is what we call this process translation. So remember, as we said before, the information has been transcribed from DNA into RNA. The RNA, specifically the messenger or mRNA, is the one bringing that information in the form of a triplet code. We call them codons. And now the cell has to assemble the corresponding polypeptide chain. Okay, let's go here. So this process is going to happen in 3 steps. The first step is called initiation. You're going to form a complex between the ribosome, remember, that is the structure that contains several different proteins as well as ribosomal or rRNA, the messenger RNA, and then a specific transfer or tRNA, and they're going to form a complex. The next stage is called elongation. At this stage, basically, amino acids are going to be joined to the growing polypeptide chain, and it's going to be growing just like as it reads the genetic code. tRNA molecules with their corresponding anticodons are coming in and linking the amino acids in the growing chain. Termination is when the proteins finish synthesizing. The ribosome mRNA and tRNA complex dissociate as they find one of those 3 termination or ending codons that I mentioned in the previous presentation. So here is a sketch. This is a very small picture. I'm sorry, but this is what I have. So hopefully you can pause or maybe look at the picture in your PowerPoint handout and look a little bit more. But you can see here where as the mRNA shows this AUG sequence here, that tells the synthesis mechanism to bring in a transfer RNA that has the anticodon for that, and that is the one that has the amino acid methionine. As the system assembles and continues, it starts adding other and other amino acids. So you can see here where the phenylalanine amino acid was added, and then it continues adding other ones and other ones and other ones. As each amino acid is added, the polypeptide chain starts kind of like uh, dissociating itself from the complex, and you start seeing this string of amino acids kind of like weaving their way out of the ribosome, where only maybe 1 or 2 of them are left there. Um, the system has to kind of shift, and we call this translocation. So in other words, every time you're going to add a new amino acid, these guys have to kind of shift over a little bit because the ribosome holds essentially 2 amino acids in there at a time. At the end of it all, you're going to reach some kind of a stop codon, in this case, UAA, and that's going to tell the system that it's time to stop. And so the ribosomes and the mRNA and the tRNAs are going to dissociate, and the polypeptide is going to be either used right there or transported to other parts of the cell or perhaps to the exterior of the cell. I'm going to ask you at this time to go to your Canvas assignment page. This is where the lectures PowerPoints are, and there should be in there a link to a video that has an animated sequence of, an animation essentially about the whole process of translation, and it shows us in a way that I possibly can't. Once more, Because of copyright issues, I'm not included it in this video presentation, but you can go there and watch it. Please pause this video while you do that, and then please come back. So one of the major aspects of the transfer of genetic information is how does it get controlled? In other words, if every cell of the organism has essentially um, the potential for the same information, how do some cells in our body become skin? How do some become muscle? How do some become, intestinal tissue? You know what I'm saying? There has to be a way of controlling that information. There also has to be a way of controlling how much of particular protein gets made. For example, if the system, the organism needs insulin, which is a protein, but it wants to say, we don't need this much or we need more. How does the system tell the protein synthesis mechanisms to either speed up or slow down the production of this particular protein? So we don't have time here to go into all the details, but I'm going to give you some general ideas about how gene expression is controlled. The DNA of each living thing contains thousands of genes, and again, these are not going to be continually read to make proteins. This is what we call gene expression. So we say that when a gene is actively making protein, we say it's being expressed. The production of unneeded proteins would be an inefficient use of resources. And so, by these mechanisms of control of gene expression, we can prevent the manufacture of unwanted proteins. Now, as we look back on the different stages of the process, you can think of different points at which control could be exerted. For example, you could simply go to the transcription of mRNA and simply selectively turn some genes on and some genes off. Or you could simply amplify transcription, say, let's make one gene more active than other ones, and so make more messengers of a particular gene than other ones. You could also, once the mRNA is in the cytoplasm, you could have a mechanism that selectively translates one mRNA more efficiently than others. And so you make more of that protein and less of the other ones. Or you could simply keep the mRNA around for a longer period of time. And so it gets translated for a longer period. We'll call this prolonged translation. So as you can go back and think about the different stages that we talked about, transcription and translation. And you can think of what would be some points at which the process could be controlled. One of the earliest mechanisms discovered in the field of molecular genetics was what's called the operon system. And this was discovered in bacteria because they were shown that when they were presented with lactose as its only carbon and energy source, they could literally turn on specific genes for digesting lactose that they didn't use otherwise. And the mechanism simply consisted of a set of genes. Here, they're simply called LAC-Z, LAC-Y, and LAC-A. And then there was a sequence here called PLAC, which is the promoter for those genes. And then over here was a section that was called a repressor. It's called LAC-I. So what was found was that gene in particular transcribed into a messenger that they got translated into what's called a repressor protein. That repressor protein could bind to something here in the DNA called the operator sequences, and it would block RNA polymerase from transcribing these genes that were important. Now, in the presence of lactose in the medium, what would happen is a byproduct of lactose would come in and inactivate that repressor. As a consequence, the repressor would fall off these operator sites and now unblock the RNA polymerase, which would then would be able to transcribe all of these genes that made essentially the proteins or enzymes involved in the digestion of lactose. Uh, then you had a mechanism when once lactose was removed, this repressor got active again and would again block these enzymes from being transcribed and translated, and so the cell would revert to its normal use of other metabolites in the growth medium. It's a very elegant system. It was discovered purely by microbiology and genetics methods, and later on, biochemistry came in and was able to identify all these different molecular components. This is a very simple system, and there are several systems in higher cells than bacteria that operate similarly to that. Now, one of the concerns that we can deal with is the issue of mutation. Let's talk about that for a moment. As we said, the replication system in the cell is in charge of making sure that anytime DNA is replicated, you preserve a valid copy of the original information. But as we said, every now and then you can have a change in the primary structure. In other words, the sequence of nucleotide residues in DNA. And that kind of change, we call it a mutation. Mutations can come in different ways. It could be the switching of a base pair, or it could be addition or deletion of base pairs. There are different kinds of mutations. And there are some common causes of mutations. Of course, there's always a possibility of an error in the replication process that didn't get caught by either the proofreading function of DNA polymerase or by the action of the various DNA repair enzymes. There are also environmental agents that we call mutagens, and these are essentially elements, chemicals, radiation, aspects of the environment that can cause these mutations. So this could be, for example, UV radiation, X-rays, nuclear radiation, and also possibly some chemicals. This is a chromosome map, so to speak. As we said, the human genome consists of, the typical cells have 23 pairs of chromosomes. And of course, they go from 1 to 22. And then in the case of a female organism, you'll have two copies of the so-called X chromosome. But in the male cell, you'll have a X and a Y chromosome. Scientists have been able to map many common inherited diseases to specific mutations in the map of these chromosomes. So you can map them to specific chromosomes and then to specific sections of these chromosomes. For example, here is a very well-known disease, sickle cell anemia. As you can see, it maps to a particular section of chromosome number 11. Some of these are not as straightforward as they might seem here. Some of them, for example, breast cancer, there are several genes that seem to be associated with that condition. It is not specifically known all of the functions of these genes, and this is one of the major areas of research if you're ever interested in going into any kind of research. Of course, this is one of the perennial hot topics is, can we find out not only what is the genetic cause of specific diseases, but what is specifically the molecular events or structures that are affected by those mutations. I invite you to look at this map later on. You might find some interesting stuff in it. Now, of course, leave it to human beings to be able to take matters in their own hands and do things. Starting first of all, of course, going back in the ages, we have known that we can manipulate the transfer of genetic traits by a process we call breeding. So we've known how to breed crops of corn or wheat. We know that we can breed horses and different animals to produce or maintain certain traits that we're looking in them. So we've been able to do that. But in the 20th century, the biochemical tools became available where we could actually go in and chemically manipulate DNA and generate what is called traditionally recombinant DNA. So, in a recombinant DNA, what you do is you take two or more DNA segments that are typically not found together in nature. In other words, what you do is you basically take, let's say, DNA from a bacterium and you put in DNA from a higher organism. And this is done by specific enzymes that are called restriction enzymes. We found out that there are enzymes available that can cut DNA at very specific sites and generate what we call sticky ends. Call them here. In other words, little single-stranded leftover DNA that now has available bases to base pair. And so if I take a circular piece of DNA that is found in some bacteria, called a plasmid, and I cut it with a particular enzyme that makes a single specific cut in there. And then I take DNA from another organism and I cut it with that same enzyme. What that means is that it's going to generate fragments that have these sticky ends that can attach specifically, complementarily to the sticky ends of the plasmid. And now what I've done is I've generated a recombinant plasmid. Most of the DNA in this circle is generated from original bacterial DNA, but then you have this segment here that is generated from a higher organism. So, for example, one of the first genes, or very important genes that were made this way, was the gene for insulin. You could actually put it into bacteria and then grow these vats full of bacterial cultures that could, dish out, you know, a lot of insulin that can then be purified and used to replace the insulin that used to be taken out from, uh, you know, from animals, from animal samples before. Uh, the product originally, I think, was called Humulin, and I think it's still around. This is a very interesting experiment that was done way back when. So essentially, there are some jellyfish that generate this green fluorescent protein. And these jellyfish make this protein. And so what scientists did was they isolated that gene and were able to put it into a worm called, oh gosh, C. elegans. It's like an earthworm. And what was interesting was that when they transfer the gene, they also transfer its regulatory section. So that now you could actually see where these things lit up, and you can see a lot of activity here in the, I think this is called the proboscis or the mouth of the worm, and you can see a lot of activity there. And so this was done essentially, what you did was you attached, let's say, a promoter sequence to this GFP protein. And so whenever that promoter becomes active, it's going to make this protein that can be easily identified because of its fluorescence. And that way you can tell at some stage in development what areas of, in this case, the worm are very active because they light up with this green fluorescent protein. Again, it sounds like science fiction, guys, but this was actually done. And nowadays, many other procedures can be done this way, and scientists use them to analyze, when and where certain genes become active during the development of an organism. Of course, another important thing is chemotherapy. So remember that in cancer, right, in oncogenesis, what happens is that certain cells in certain tissues just basically lose their control and they start duplicating out of whack. So whatever mechanism was originally controlling the cells in our tissues suddenly just went awry and these cells start reproducing out of control. And of course, the cells they produce are non-functional and so they obstruct the functioning of other important tissues. So chemotherapy is a method that is used essentially to interfere with DNA replication. Here's the idea. If you can find a way that blocks DNA replication, but it basically targets cells that are replicating a lot faster, predominantly over normal cells. Then you can target that treatment and essentially prevent the cancer cells, which are growing very fast, from growing while attempting to keep the normal cells from getting killed by this chemical. There are different kinds of chemicals in chemotherapy. Some of them simply go in and interfere with base pairing. So these are things that basically modify bases. This is called alkylating agents. They basically modify bases so they can no longer pair. Of course, a cell that is replicating very actively is more likely to incorporate this chemical and have its bases modified in this way. There are other chemical treatments that prevent the uncoiling. These are topoisomerase inhibitors. Remember, in order for DNA to replicate, it has to open up, right, into this replication loop. Well, if you could block that, you might be able to prevent those cells from continuing to replicate. Other ones affect other functions that have not directly related to replication, but the actual cell division. And so, for example, there are some kinds of chemicals that prevent the formation and function of the microtubules. These are the cellular structures that essentially allow the chromosomes to move from away from each other and into the new cells. And then another time is the ones that block essentially the formation of nucleic acids. These are different kinds of chemicals that interfere with the function. Again, all of these chemicals, their objective is that if you can give them in the right amount, you will end up affecting only those cells that are actively replicating and killing them, but leaving the majority of normal cells healthy. This is why some of the side effects of chemotherapy, for example, the loss of hair, the shedding of the lining of the GI tract, which produces a lot of, nausea and vomiting. That's because those are tissues that are by themselves normally replicating at a higher rate, and so they end up incorporating, unfortunately, some of these chemicals, and these are some of the side effects of chemotherapy. And that is as far as I'm going to go. If we have time in our class, I want to talk about other topics of more recent uh, uh, developments. Those of you growing in this generation have probably heard the term CRISPR. This is a very highly specific way of modifying DNA sequences. Also, we can now synthesize in lab specific sequences of RNA and keep them stable and inject them into cells. This is the basis for the so-called mRNA vaccines, such as the ones developed against COVID during the pandemic of a few years ago. I wish I had time to talk about these things, but unfortunately, we don't. And these are very sophisticated topics, but feel free to come by and ask me about it, and I can give you a few ideas of where this is going. Thank you so much. I hope these presentations help you in your study, and I'll see you soon.